The first system we'll be talking about in 139 is going to be the cardiovascular system. And you'll see we're going to break that system down and take it one piece at a time. The first thing we have to get into is blood and blood composition. Now, admittedly, some of the topics we're going to be talking about with blood we're going to introduce now, with the intention of looking at them in much greater detail once we get to some other systems. So we'll talk about things like blood gases and blood pH, but we won't fully get into those and cover those topics until we get done with respiration and renal. We're going to talk about immune function, white blood cell function, but we won't fully engage that topic until we get to the immune system as a whole. So some of this introductory topic we're very much going to be in the now, and this is when we're going to get to the nuts and bolts of it. However, other topics will be more of an introduction as we sit here in the cardiovascular system because of the overlap the cardiovascular system happens to have with these other body systems. When we look at blood and blood composition, it's important to go back and remember what connective tissue is, because blood is a type of connective tissue. You need to understand the breakdown of blood, its fluid components, its cellular components, its proteinous components. We're going to look at blood typing, one of the major topics in blood and blood composition. And you'll be introduced to that one in the lab as well. And eventually we'll get into some blood clotting and at least an introduction to some immune function. When we remember back to connective tissue, in A&P 1, we discussed connective tissue from a connective tissue proper standpoint, where we looked at loose connective tissue, dense regular, dense irregular, and we would have covered that under our tissue types. We later on expanded and looked at a more specific type of connective tissue and how it applied to the skeletal system when we looked at different types of cartilages. But as a whole, connective tissue is going to be one of the most varied, physically appearing tissues in the human body. It can be an absolute solid, rigid, like we see in bone, and it can be a liquid, like we see with blood and lymphatic fluid. And in between those two extremes, we're going to get various degrees of solidity. We see very pliable, very soft connective tissues, like we see with areolar connective tissue, various cartilages. Now, regardless of those different types of physical appearances, blood, bone, and everywhere in between, we still see connective tissue have the same basic composition. There has to be a cellular component. Now, that doesn't mean that the cells we see in blood, cartilage, and bone are the same type of cell, but all across that spectrum, we have a cellular component. There has to be a fibrous component. And just like the cells, it doesn't mean the fibers that we see in one type of connective tissue will be exactly the same type of fibers we see in another type of connective tissue. But right across that spectrum of appearances, blood, bone, and everywhere in between, we do see a fibrous component to all of those different subcategories of connective tissues. And then holding it all together, suspending those first two components, we see this ground substance matrix. Now, it is going to be that matrix that will give the vast physical characteristics to these types of tissues. I like to look at an old analogy I've been using for a while of a 1970s Jell-O dessert. You would see them make Jell-O and put it in this bunt pan till it firms up, and you tip it upside down, and you get this Jell-O mold. Well, picture you're making that Jell-O dessert, and you're going to put bits of fruit in it. The fruit's going to represent the cellular component. Now, in addition to the fruit, we're also going to put spaghetti noodles in it. The noodles representing the fibers that are going to be part of that connective tissue. So you put that in the fridge and you let it firm up and you take it back out, and you can see the kind of model that we're talking about now. Cells and fibers suspended in this ground substance, Jell-O. Now, if we take that Jell-O mold and we chuck it in the freezer, it's going to get rock hard bone. We take it out of the freezer and let it reach room temperature again. And it's going to be very pliable and very jiggly. Cartilage, semi-solids. We take that same Jello mold now and we warm it up again, and it goes right back to being a liquid. There's going to be the physical appearance that we get with a blood and lymphatic fluid type of connective tissue. But right across that spectrum, we didn't lose the fibers, we didn't lose the cells. We just changed the physical characteristics of the ground substance. And here you can see more of a rendition of a cellular structure, with the various fibers running through that tissue, our various types of cells, depending on the type of connective tissue we're talking about, and all the while suspended in this ground substance matrix, that taupe background. We had loose connective tissue, and we had dense connective tissue. When you hear terms like that, it's referring to the density of the fibers. And you can see when we look at this loose connective tissue, we see fibers, but we see a lot of that ground substance in the background. A low fiber content. That's what gives loose connective tissue. It's more rigidity. Now, blood, to stay on topic, is going to be a loose type of connective tissue. We see proteins such as fibrin, fibrinogen, that play a role in blood clotting. We see proteins like albumin, small, non-fibrous proteins circulating in that blood. We see various immune complexes, immunoglobulins circulating in that liquefied matrix. But overall, we see blood have a high ground substance to proteinous fibrous structure. When we look at the overall functions of blood, it can first appear to be a little bit convoluted and overlapping with some other functions. So when we say transportation, what exactly is being transported? We'll talk a lot about gases being transported throughout the blood, carbon dioxide and oxygen in particular. Nutrients, waste products. When we talk about regulation, the main thing we're going to see regulated by the blood content is going to be blood pressure. Changes in diameter of blood vessels, changes in heart contractile strength and rate, dictating the pressure of the blood within the vessel. But we also see blood play a role in fluid volume regulation. We'll talk about fluids shifting into and out of the tissues, going from the capillaries into the tissues and vice versa. We'll also talk about blood volume being regulated by the urinary system. And then protection. Here's we kind of get more into an immune response. When we look at the overall structure of the cardiovascular system, we have the heart, the one that's going to get the most recognition. We'll spend several weeks talking about the heart. It's a muscular organ that when contracts, develops pressure on the fluid blood inside, driving that blood through a network of blood vessels. Now, in lecture, you won't have to be responsible for any naming of blood vessels outside of what we're going to call the great vessels. The large arteries and large veins that are directly associated with the heart itself. Once we get into heart anatomy, you'll definitely need to know those. In lab, you'll be more responsible for naming specific arteries and veins. For lecture, I want you to just get a feel for directional blood flow. We have our blood vessels. The ones that leave the heart are going to be the arteries. You will see other definitions maybe thrown about where they say arteries carry oxygenated blood, and that's not necessarily always the case. There's a couple of very good exceptions to that we'll see. Arteries carry blood away from the heart. Large. Those large arteries will divide into smaller arteries. Those smaller arteries will divide into smaller arteries again. They keep dividing and getting smaller until we eventually get the smallest arteries, the arterioles. Those are going to be the ones that we'll discuss as having the greatest ability to dilate and constrict. Because of that ability, they're going to be the ones directly involved in a lot of blood pressure regulation. They have the greatest percentage of ability to change its diameter. Once blood goes through the arterioles, now they find themselves in the capillaries. And we'll be referring to those as the functional blood vessel in this network. This is the only point in which the cardiovascular system can exchange materials with the tissues. These capillaries are porous, so they allow fluid to leave the blood vessel and go into the tissues, and vice versa, carrying that bidirectional exchange of nutrients and oxygen getting into the tissues where the cells are, and waste products and carbon dioxide being brought back in the opposite direction. Blood that makes its way through the capillary will find its way into the smallest of veins, the venules. Those small veins will converge now. Two small veins converge into a larger vein. Those two larger veins converge into even larger veins again. So eventually, we get to the largest veins, and now we're right back at the heart again. One complete circulatory loop. Once we get done with the cardiovascular system, we're going to come right back and look at the lymphatic system, because it really is the other half of our fluid volume. We will be discussing blood and blood volume as being major regulators of blood pressure. But as we shift this fluid into and out of a capillary, we can never discount the percentage of fluid that's imbalanced. We lose more fluid from the capillary than we bring it back. There's where our lymphatic discussion comes in. Lymphatic vessels make up for then account for that additional fluid loss from the capillary and recoup that fluid back into the circulatory system. Now, when we look at blood composition, we can break it down in a couple different ways. The most well-known components are going to be our cells, and for our purposes, we're going to refer to them as the formed elements. Red blood cells are erythrocytes, white blood cells are leukocytes, or platelets, or thrombocytes. Now, we don't refer to these as the blood cells per se, because the platelets are not a cell. They're a cellular fragment. So to group all those together, we refer to them as the formed elements. With the red blood cells, as you'll see, get taxed with moving gases throughout the blood primarily. These will be the cells we'll talk about for blood typing. The white blood cells, we'll look at the structure of them today. We'll introduce some of the function. But these will be the ones you'll come to know as having the immune response, antibody production, phagocytosis of a foreign invader. We'll finish up that topic once we get to immune. And platelets are going to be known for their role in blood clotting or hemostasis. Plasma will be the part of blood minus the formed elements. So it'll be that liquid component plus all the proteins. The albumins that play a role in fluid shift. The globulins that play a role in immune function. And the fibrinogen that plays a role in blood clotting. Now, if we pull out the clotting factors out of that plasma, now we have serum. And serum is what you may have heard referred to when people go and they donate plasma. You leave the clotting factors in there when you're donating plasma. Now, because of all those components can have varied densities, when we take blood out of the body, we centrifuge it, spin it down, we will see the more dense part of the blood sink to the bottom of the test tube, while the less dense structures seem to float on the top, if you will. What will sink to the bottom will largely be our red blood cell layer. On top will be plasma, pretty much water with some proteins. And in the middle, we'll get our buffy coat. And there's where we'll see our white blood cell or leukocyte components centrifuge out at. small percentage of blood overall compared to the other two layers. Now, of course, within each of those three major layers, we have other sub-layers, if you will, because our white blood cells will look at different types. They can tend to separate out over that buffy coat. The components that we see in the plasma aren't all having the same density, so they can be found at different parts of that layer. When we look at the different types of blood cells, I want you to be responsible for all the aspects of red blood cells for this section. Structure, function. For the white blood cells, for now, identify the structure, because it's going to be useful for you in lab, so we're going to reinforce that here. And major functions. We're not going to get into in-depth immune function though until we get to the immune system. Platelets, we're going to be discussing more from a functional standpoint. Once we get to hemostasis, we'll look at platelet plug formation and blood clotting. Here's what a typical slide may look like when you see it in lab. Now, when we looked at that layers that form once we centrifuge blood, you saw that the red blood cells were a major component, very prominent layer. And that's what you see. Mostly red blood cells. You'll see the occasional white blood cell. And to be honest with you, on most of your slides that you'll see in lab, in any one view, you probably won't see 4 different white blood cells on the same slide. They'll be less numerous, but I guess they're brought together in this one for illustration purposes. And up top on the left, you'll see platelets. You can see by looking at the platelets that they're rather small compared to the other formed elements. Remember, those are cellular fragments, not even referred to as cells. But here's our red blood cell. It's a biconcave structure without a nucleus. And when we say biconcave, when you look at the top of that diagram, see how we get concave curvature on both sides of that red blood cell. It's like we took a round piece of dough and pinched it. We see that exhibited on our slide when we look at a red blood cell and we see this darker outer ring and a more transparent center. And that is simply because cells thicker around the rim. It's thinner in the middle, more light can pass through it. This is an extremely flexible cell, and that plays into its function very, very nicely. This is going to be the primary transporter for our gases, as we talked about. We get a protein called hemoglobin, an iron-containing protein, in which we can get oxygen, carbon dioxide bonded to it. The flexibility of the membrane is going to be important for its function. As we said, if you look at this video, you see the numerous red blood cells. Remember, it's a major component of blood, making its way through these blood vessel networks, bumping off of each other, taking very close bends, converging with other capillaries, and you're having red blood cells collide. The flexibility of the membrane is going to allow that red blood cell to undergo that form of transport. Lower risk of damage. But the more significant function of that flexibility will be for its ability to transport. Way back in 137, AMP1, you discussed diffusion. Now, most of that discussion was diffusion into and out of a cell. But you surely discussed diffusion in a more basic sense as well, like taking a pitcher of Kool-Aid and putting a bunch of crystals in there, and you got that concentration gradient. Come back over a period of time, and now the crystals are more evenly dispersed out as you reach that equilibrium. There were a couple of principles that dictated the rate of diffusion. One of those was distance. Now, we would have brushed that off in AFE 1. We said that the distance wasn't really applicable to that particular context of topic because, again, we were talking about diffusion into and out of the cell. So the distance would have been the thickness of the membrane, which we would not have seen as being variable to any significant extent from one type of cell to the next. So the distance would have been constant for any type of diffusion we were discussing at the time. But now we're talking about diffusion from the red blood cell across the red blood cell membrane, through the plasma, through the capillary wall, and then ultimately into the tissues. So we're talking about what could potentially be a much more variable distance. So while it might not have come into play for diffusion into and out of a cell, it certainly comes into play when we discuss diffusion of gases from a red blood cell and out of the capillary. To make sure we cut down on this distance, we want our red blood cell wall to sit as tight as possible up against the wall of the capillary. Because again, the wall of the red blood cell isn't going to be variable. The wall of the capillary isn't going to be variable. The variable that we have the potential to see here is the space between the red blood cell and the capillary, that plasma in between. So if we can make the red blood cell larger than the capillary, but give that red blood cell a very flexible membrane, so as it hits the capillary, it can deform and squeeze its way through. In order for it to do that, the walls of the red blood cell had to have been tight up against the walls of the capillary, minimizing that space, minimizing the distance, therefore making the rate of diffusion from the red blood cell and ultimately into the tissues as minimal and high a rate as absolutely possible. Let's look at that one more time. And you can see how the red blood cell deforms in order to make its way through that capillary. And that's going to be directly relating to the flexibility of that red blood cell membrane. Oxygen is not going to be very soluble in plasma. That's why the hemoglobin proteins and its binding affinity is going to help that out. Now, we'll also see hemoglobin have an affinity to bind to carbon dioxide, hydrogen ions, carbon monoxide in particular. And these are topics that we'll bring up once we do respiratory. People with carbon monoxide poisoning. And how we see oxygen and carbon dioxide have this dissociation curve. How does the hemoglobin have the ability to pick up oxygen at the lungs and bind oxygen at that site, but then get to the tissues and kick that oxygen out? So we'll talk about those principles once we get to respiratory. But again, hemoglobin is a 4-protein chain, each chain having this iron-centered heme molecule. That's where we will get our binding sites for our oxygen. Now, blood typing, we said earlier that it's going to be a major topic for lecture. It's also a major topic for lab. So we're going to look at that a little bit more in depth so we can really drive home those concepts. When we look at blood typing, we're even starting to introduce a little bit of immune function a little bit, because we're going to use some terms that we're going to use for immune. Antibodies and antigens. Antigens are proteins that sit on a cell wall. If you produce an antibody that's going to attack a bacteria that's invaded the body, It's going to recognize proteins on that bacterial cell wall. It's going to recognize antigens on that cell wall. Well, all your cells have proteins. In this case, on the red blood cell, we get two particular proteins that show up on our red blood cell. Those are antigens. We have the A protein, the A antigen, and we have the B protein and the B antigen. And based on the presence or absence of those combination of proteins, we get our ABO system. If you know your blood type, it may be A, it may be B. If you're type A blood, it means you have the type A antigen on your red blood cell. If you're type B, that implies you have the type B protein on your red blood cell. Now, you can have a case where you have neither. Your particular red blood cells lacks the A and it lacks the B, in which case we give it the O designation. You would be type O blood. But there are cases where you could have both antigens. Your red blood cells may have the A and the B, in which case your blood type would be AB. Now, most students don't have a problem with the antigen portion of blood typing. As long as they know what the antigen as a term means, they pretty much nail those. Where students get tripped up is that they don't realize that this ABO system is a complementary system. It doesn't just rely on the antigens for its composition, even though it's the antigens that give the bulk of the naming. In addition to the antigens, we have a complementary antibody component. And I want you to always remember the tagline: you will produce antibodies for antigens that you don't have naturally. You will always produce the antibodies for the antigens that you're lacking. And that should make sense. You don't want to produce antibodies for proteins that are naturally occurring. Essentially, that's the basis of autoimmune diseases, where your own immune system starts attacking natural tissue. But you will always produce the antibodies for the antigens that you lack. Now, in this case, we're talking about IgM antibodies. Rather complex structure. Always on the lookout for the antigens that they're meant to bind to. You always produce the antibodies for the antigens that you're lacking. So in type A blood, we already nailed home that you have type A antigen. You're lacking the B. Therefore, you're going to produce anti-B antibodies. It's a complementary system. Both of those components can exist consequence-free. The A antigen, the anti-B antibody, they don't interact with each other. There's your AB system. In a reciprocal manner, if we look at type B blood, yes, you have the type B antigen, but you have the anti-A antibody. You always produce the antibody for the antigen that you're lacking. And if you follow that logic, type AB, you have both antigens. You always produce the antibody for the antigen you're lacking. You're not lacking either one. Therefore, you don't produce either of the antibodies. And in type O, you're lacking both. So you wind up producing the anti-A and the anti-B antibodies together. Any discussion that you're going to have to know about blood typing has to include your knowledge of the presence or absence of the antigens, as well as the presence or absence of their complementary antibodies. So if asked, what does type A blood consist of? It's the A antigen and the anti-B antibody. You have to include both. Now, you will see some practical questions in both lecture and lab with regard to blood typing. And there's one lab in particular that you will do where you'll get plastic trays that will have 3 wells. Plastic tray with 3 wells. And you are going to have to identify a type of blood based on the interactions you see. If we take type A blood, little vial of type A blood, and put it into a type B person, you will see the antibodies in the recipient attack the cells that have been donated. And you will get what we call an agglutination reaction. Remember, in a type A blood, we didn't have interaction between the antibodies and the antigens present. They were complementary to each other. But once we start mixing blood types, now you're mixing the antigens with the antibody that's meant to bind to it. So back to our example here, you'll be given a number of different synthetic bloods. Mr. Brown, Mrs. Smith, some naming system like that. It doesn't tell you what type of blood they have. You will take one type, Mr. Brown, for example, and you will take Mr. Brown's blood and you will put it in all 3 wells. Then you will get synthetic antibodies. You will get a little vial of anti-A antibodies, and you will get a little vial of anti-B antibodies. Now, this is where the named wells come into play. You have an A well and you have a B well. Now, both of these have the same type of blood, remember? You will take the anti-A antibody and put it in well A. You will take the anti-B antibody and put it in well B. Now, what I want you to do is get a logical system. Treat it like a math problem. Back when you were studying basic math, you didn't have to memorize every single type of equation they could give you. You understood the pattern on how the problem needed to be solved. Then regardless of the question they gave you, you applied that pattern to it, and you got the correct answer. We're going to give you a pattern, a system to work in here. Remember, A and B, those are our options here. Those are the only 2 antigens that could be present, right? So we put our synthetic antibodies into each of those wells, and lo and behold, this one gets cloudy. Now, in the lab, it may get cloudy, it may get clumpy, you may see very fine hair-like structures forming through it. All of those will indicate that an agglutination reaction took place. So here we have a cloudy well A, but we have a clear well B. Here indicates that the antibodies interacted with an antigen. Here, no agglutination. So whenever you're trying to solve something like this, jot down these 2 letters. Did we get a reaction in the B antibody, in the B well? No. Done. Therefore, this has to be type A blood. Now suppose this one also remained clear. You put the synthetic A antibodies here, you put the synthetic anti-B antibodies there, yet they both remain clear. Did we have the B? Nope. Do we get any reaction with any A antigens present? No. What do we call the blood type that's absent of both? Now you can designate it as type O blood. This is going to be one of the first topics where you will see a spotlight session, where we'll be going through that topic in just a little bit more finer detail, and it'll be reinforced in lab as well. Along with this AVO system, you will see a couple of terms: universal donor and universal recipient. Type O is going to be considered our universal donor. Type AB is going to be considered our universal recipient. Now picture what that means. When I said before that we take type A blood and we put a small vial of type A blood into type B, we see the antibodies in the recipient attack the antigens on the donated cells. We can call type O a universal donor because we can put type O into any of the other blood types because it doesn't have any antigens to attack. So if we put type O into type A, the A antibodies have no antigens to bind to. If we put type O into type B blood, the anti-A antibodies have no antigens to bind to, and so on. Type AB is the universal recipient because it does not have any occurring antibodies. So whatever type of cell we put into type AB blood, there are no antibodies to attack the cells that are being donated. Now those terms, universal donor and universal recipient, it's important to recognize that these are volume dependent. You can't take buckets of type O blood and force it into a recipient without consequence. And the main reason for that is because, remember, when you are donating type O blood, you're not just donating the cells with the antigens or lack of antigens, but you're also donating the complementary antibodies. If you're a type A person and you will need a little bit of blood, you can put the O into type A, and the O cells that are donated would be okay because there's no antigens on there which to be attacked. However, if we took a large volume of O, let's say this type A person lost a massive amount of blood, and we tried to replace that with type O blood, it still remains true that the donated cells would be okay. Because again, there's still no antigens on those cells. However, in that case, we wouldn't be worried about the recipient rejecting the donor. We would essentially be worried about the donor rejecting the recipient, because the large amounts of antibodies that would go with our donated O cells would attack what little blood that was still left in the type A person. So those are going to be volume-dependent terms, universal donor and universal recipient. Now, one thing to clear up is that we are talking about blood-blood interactions here. We're not talking about what you might go to a hospital in an industrialized country and get a blood transfusion. We are talking about what would happen if we mixed one person's blood with another person's blood. In a hospital setting, what you often see is packed cells. So if you're donating type O blood now, antibodies may have been removed. So all you're getting is type O cells being donated, not type O blood. So it's important to make that distinction. Now we have one more component to this ABO system. And some of you, if you're already familiar with the blood type, you may have already been wondering this along the way. Because you may not be type A. You'll be type A positive, or you'll be type A negative. There has to be a designator, positive or negative, associated with the ABO system. That is a third antigen. So we had the A, we had the B, we had combinations of those to give us our type A, type B, type AB, and type O. The third antigen is our rhesus factor. If you are positive, it implies that this antigen is present. If you are negative, it implies that that antigen is absent. Now, please don't forget to write the negative there. We see students all the time, if they've worked their way through an example and they've figured out that it's A positive, they know to write the positive in there. But if that antigen has to be absent, then you still have to actually write the negative. Just writing type A is not going to be the same as writing A negative. So now we go back and look at our example again to finish off what you will actually see in the lab. Now you just got a third antigen to take into account and a third type of synthetic antibody. So now when you're writing out your example, I want you to start with that. That way you'll never forget the negative sign if it turns out to be a type or a negative type blood. When you start your problem, write an A, write a B, write a positive sign and a negative sign. Write it all out there. You will take your blood, your unknown blood type, put it in all three wells. You will take your three types of synthetic antibodies. You will take your anti-A antibody and put it in the A well. You'll take your anti-B antibody and put it in the B well. And now you will take your anti-rhesus antibody and put it in the RH well. So move the tray around a little bit. You'll come back after a few minutes and try and observe any wells for agglutination. Now this time, let's say we got some cloudiness here, and then we got some hair-like structures throughout the rhesus factor. Let's look at our tray now, go through our system, and figure out what blood type we have. Did we see any indication that there was a type A antigen? Well, stay clear, no agglutination, the antibodies had nothing to attack, therefore the antigen was absent. Get rid of it. So you know it's a type B at this point, because we did get agglutination here, indicating the antigen is present. Now we look at our rhesus factor. We see some agglutination indicating that this antigen was present. Now we get rid of the negative, because the positive implies that the antigen was here. There is your blood type for that unknown. So in lab, you'll do this as an exercise. You go through some different blood types. But on a lab exam or on a lecture exam, you may get just a picture of the well and have to identify what blood type we get. You may get a question that says you perform this experiment and you get agglutination apparent only in the A well. What type of blood do we have? Again, don't be afraid to draw this out. Get your four indicators up here and then get rid of them as the details of the example implies. And what's left behind will be your blood type. Get a systematic way of doing it, and you'll be less likely to fall through the cracks on the question. Now, the antibodies we see associated with the rhesus factor are not the same antibody types we saw with our A, B, O antigens. Remember, those were IgM antibodies. The antibodies for the rhesus factor are IgG antibodies. A little different structure, but also a couple other nuances. First nuance. These antibodies can cross the placental lining. IgM could not. IgGs can. So there is going to be an implication here as far as what happens if you've got an Rh negative mother and an Rh positive fetus. The fetus being positive carries the antigens. The mom being negative has the potential to carry the antibodies. So you run the risk of the antibodies in mom crossing the placental lining because they're IgG antibodies and attacking the fetal blood tissues. Now, I said just a moment ago that mom has the potential to have the anti-Rh antibodies. There's your second nuance. For A, B, and O, if you have the A antigen, you will always have the anti-B antibody. Part and parcel, they come together. These antibodies will behave much more along the lines of what we see with immune response. So if you're an Rh negative mother, you start off life by just having red blood cells with an absence of the Rh antigen, but with no antibodies. If mom gets exposed to the antigen, as in a Rh positive fetus, then she's going to respond to that antigen by then producing the antibodies, much like you would if you were fighting off a foreign invader. So we can have our first pregnancy. Rh negative mom, Rh positive fetus. No extreme blood-blood mixing throughout the development, therefore no antibody production. However, during the birthing process, you are going to get blood-blood interactions. You are going to get the fetal antigens get exposed to mom's blood. From that point forward, because of the subsequent exposure, mom will always produce the Rh negative antibodies. So now, after the first pregnancy, is where you'll see the more typical complementary system that we saw with ABO. The absence of the antigen, the presence of the antibody. So now mom has to be careful on subsequent pregnancies. If her second fetus is Rh positive, because these antibodies can cross the placental lining, that's where you're going to get the attack. So you often see ladies who are at this high-risk scenario go on some sort of immunosuppressive medication prior to their second pregnancy to suppress the production of these anti-Rh antibodies, so they will avoid the attacking of the second pregnancy. Moving on to our next type of blood cell, we get to our white blood cell, the leukocytes. And these will be the ones that for now we're going to look at structure, we're going to look at basic function. We'll leave the in-depth immune response to our immune system. Because that's primarily what these cells are going to do. They're going to defend against foreign invading organisms, parasites, fungus, bacteria, virus. They're going to be a cleanup crew. You get injured, you get damaged, you get decayed tissue. White blood cells will play a role in debriding that tissue, part of the healing process. And you will even see a role in the identification and destruction of cancer cells. Treatment of cancer is really something that's baffled the medical community for quite some time. And for a very long time, there were only really three options. Cut the tumor out, radiate the tumor, or poison the tumor. Now, all three of those come with consequences. Sometimes tumors are in a place where you simply can't cut it out, because cutting through the healthy tissue to get to the tumor can be detrimental. Radiation has its problems. And again, you don't want to radiate vital tissue that may be in proximity to the tumor. And then we all know the destructive outcomes of chemotherapy as we try to basically poison that highly replicating tissue. But if we can find a way to manipulate your own immune system to specifically target just cancer cells, you kind of get that magic bullet now, somewhat side effect free. And there are instances where this occurs naturally in the body. Doesn't mean that we eliminate the potential of any cancers, but it is one of our inborn mechanisms to prevent tumor formation in the first place. We have 5 different types that we'll look at: neutrophils, lymphocytes, monocytes, eosinophils, and basophils, named in that order based on their prevalence in the blood. At this stage in the game, you will need to know which ones are more prevalent than others. And you get a little mnemonic based on the relative percentages that we observe and the first letter of each cell type. 60, 30, 8, 2, 0. It's less than 1. To make it rhyme, you give it an O. 60, 30, 8, 2, 0. Never let my engine blow. So you'll need to know which cells are more prominent in the blood than others. And again, we spin them out, and you will see the more dense cells within the buffy coat kind of gradient out there. You get the agranulocytes versus the granulocytes. Granulocytes, think about that name. They have a granular appearance. They're going to look spiculated, spotty. Where the agranulocytes lack that appearance. Now, we're going to go through each of those 5 types and look at distinct calling cards. I don't want you guys really to be forced with having to look at a slide in lab and work your way through 5 or 6 different characteristics to determine what type of cell you're looking at. Identify the one characteristic that's going to be your foolproof plan for these cellular identifications. First off is the neutrophils. I want you to look at the nuclei. And many neutrophils, it'll almost look like there's multiple nuclei. Now, it's called lobular nuclei. All those little bits are going to be connected, but on some cells, it'll look like there's a lot of different nuclei in there, 3, 4, 5 of them. The nuclei is not nice and round like you've seen on some other cellular drawings. It's kind of lobular, compartmentalized. When you see that type of nucleus, you know you're looking at a neutrophil. Eosinophils. They are going to have red cytoplasmic granules. Out of all of these here, it's going to be granular, and those granules are going to be a red color. Now, red might be relative depending on the stain. Might be a bright red, it might be more of a burgundy, sometimes almost a purple. Basophils begins with a B. Still granulocytes, but they're going to be very dark, almost black. B for basophil, B for black. Very dark granules. So both of these are granulocytes. If they're red, they're eosinophils. If they're very dark and black, they're basophils. One characteristic, get your naming down. Next up is going to be the lymphocytes. Now, these are typically smaller, but maybe it's the only cell you see on the slide, so you're not going to get a relative size. When you are looking at a lymphocyte, the description I always put with this is that it looks really clean. Number one, it's agranular, so it doesn't have the spots. And number two, the nucleus inside is large relative to the size of the cell. So it's a very clean looking cell. And sometimes on these, all you'll see This nucleus. Non-granular, nice, smooth-looking, purplish stained nucleus. It lacks the granular structure that you see in any of the preceding ones. And then finally, the monocyte. Very characteristic shaped nucleus. Horseshoe, kidney bean shape, big fat nucleus that takes on that characteristic C shape. So get those one characteristic down, make yourself up cue cards with a picture of one on one side and the description and function on the other. Neutrophils are phagocytic specialists responding to bacterial infection. Now you'll have to think back to AMP1 for phagocytosis. We will see a small invading organism get engulfed by a white blood cell. Once inside, there will be enzymes to break down whatever it just got engulfed. Sometimes those vesicles with the enzymes can undergo what we call a programmed cell death. Rather than bringing the bacteria into the cell, we inject the enzymes into the extracellular fluid by exocytosis, kind of a drive-by killing for a foreign invader. You get a programmed cell death where the cell lyses, spilling those vacuoles out into the tissue contents. Lymphocytes are what we're going to focus on most when we do immune, because this is what we see for our adaptive immune responses. Right, as I record this lecture, we're still neck deep in our COVID pandemic. The vaccines just got released and started to get administered last week, maybe the week before. You expose the body to the antigen. You respond by producing antibodies, our humoral response. That'll be your B cell type of lymphocytes. The T cells are more the hand-to-hand combat, doing more cell-mediated immunity, more phagocytosis, more up-close and personal fighting off that invader. When we do immune, we'll talk about the natural killer cells. My favorite name of all cells. They have ways to identify stressed cells that might even be lacking other types of markers. They don't need to have the same sort of activation sequence that a B or a T cell might need in order to tackle a cell that's been infected. The monocytes are also phagocytic specialists. And they mature in a little bit of a different way. They're going to be released from the bone marrow, circulate throughout the body briefly, then they lodge in the tissues where they finished their maturation. Eosinophils are going to be more associated with parasitic infections and allergic responses. They'll attach to a parasite, and just like we saw with the exocytosis from the neutrophils, they'll secrete chemicals that will attack the parasite. And last up, we have our basophils. Allergic reactions, vasodilators. We'll see the vesicles, those granules in a basophil, contain heparin and histamine. Heparin is going to be an anticoagulant. Histamine is going to be a vasodilator, so the blood vessels in the area bring more blood into the tissues. So people that are having an allergic reaction will typically see swelling, redness, an increase in fluid in that area. And the last formed element is going to be our platelets, and plays a whole specific role in blood clotting, hemostasis. Not actual cells, but they are cell fragments that come from a much larger cell called a megakaryocyte. These megakaryocytes stay in the bone marrow, fragment off platelets that go into circulation. So we see our hemocytoblast that we'll talk about in the next section, develop into our megakaryoblast, and ultimately a mature megakaryocyte staying in the bone marrow the whole time, chipping off these much smaller fragmented platelets that then go into circulation for roles in blood clotting and platelet plug formation. We see synthesis of platelets increase with the hormone thrombopoietin. In cases of vascular damage, this hormone will be released, platelet formation will step up its game, and we will set up that system on the right foot to repair damage in that blood vessel. And the last component I want to mention is the plasma proteins. Now, we will talk about different ones of these throughout the course, depending on where we are. The first ones we'll get to are going to be the fibrinogen. That's going to be the type of protein that's involved in our hemostasis, blood clotting. We see these proteins turn into an insoluble fibrous net, which then catch blood cells as they go past, forming a clot. We'll talk about the globulins once we get to immune function. And we'll talk about albumin when we talk about fluid shift into and out of a capillary and blood pressure. The albumin is a very prominent protein, very small protein. And if you remember osmosis from ANP1, osmosis was all about the particle count. That's going to be albumin. As you become dehydrated, the concentration of albumin goes up, increasing the osmolarity of the blood, triggering our feedback mechanisms to ingest more water and retain more fluid from the kidneys. So different proteins will pop up along our discussion for different topics. But at this point, once we get to it, it will be important for you to go back and look over osmosis and its principles as well. Because if we had all the albumin concentrated on one side of a membrane, we will see water move in the direction of that hypertonic environment. So for our blood composition, it's important to know the function and structures of all of our formed elements. You should know the functions of the white blood cells in particular. And focus on blood typing. Use the system that we illustrated earlier. And once we do some special sessions, some review sessions, we'll even go over that a little bit more, different examples, to be able to identify an unknown blood type. Now that we've reached the end of this particular section, I want to reiterate something that we would have covered in our course introduction. For a lot of people, as we just went through this material, it might have been difficult to grasp at the speed we were covering it. And I'm going to try and stay optimistic as we go through this course, but this course won't be slowing down anytime soon. It's a lot of material in a very short period of time. What I'll encourage you to do, and again, this would have been gone over in our introductory section as well, is if you go to the end of your syllabus, you will see chapters associated with each topic. The PowerPoints will be available as course notes on Blackboard. Before you sit down and watch the lecture, I want you to read the chapters associated with that material, and I want you to at least preview the notes that we're going to be covering. That gives you a little bit of a base foundation of knowledge before you get to hear me talk about it. As you're watching the lecture then, I want you to be jotting down questions you have. If I say something that's not quite clear, pause it, write the question down. At the end of the lecture, you'll have any number of questions. Go back in the notes, see if you can answer those questions. If not, those are the questions that we need to address in some sort of review session. Read the textbook, chapters, preview the notes before each class, watch the lecture, develop questions from that lecture, and then come to me with those questions. And that's going to be the most efficient way you can get through this material. So if you have any questions, please let me know. We'll take it one topic at a time. Thank you.